Acid for staining for AFB purpose aided in demonstration of presence of mycobacteria AFB by microscopy examination principle. The acid was basically are surrounded by a cell wall containing mycolic acid that resists decolorization with acid. Heating helps the acid phosphate bacteria to retain the carbon friction red color, while the other takes the counter stain when stained with Ziehl-Neelsen stain. Concentration procedure aids in digestion and decontamination of any mucopurulent material present in the specimen. Acid phosphate is not specific for Mycobacterium tuberculosis. Other species in the genus Mycobacteria will also stain acid phosphate, and other organisms will occasionally stain acid phosphate. Example: Nocardia species, Rhodococcus species, and Legionella micdei. Primary samples: sputum, urine, gastric fluid, aspirate from different sites, and catheter, pus, and CSF and other body fluids. Type of containers or additives: sputum. Instruct the patient to collect deeply coughed, explorated early morning. First sample in a sterile mouth transparent plastic container for 3 consecutive days. After rinsing the mouth with water to reduce the possibility of contamination of specimen with food particles, saliva. In case of patient is not able to produce sputum, it can be induced by drinking warm water, taking mucolytic agent as a preservative prescribed by physicians. Bronchoscopic specimen to be collected by a trained personnel in a sterile container. Urine. Instruct the patient to collect early morning first urine sample complete in a sterile wide mouth container of 500 ml capacity of 3 consecutive days. Gastric fluid. Instruct to collect the fasting early morning gastric aspirate in a sterile screw capped wide mouth container for 3 consecutive days. First, instruct to collect the specimen in a sterile screw capped container or to send in aspirated syringe itself with proper sealing. CSF and other body fluids. Instructed to collect the specimen in a sterile screw capped container. Tissue, bone marrow, biopsy materials, endometrium and surgical tissue. Instructed to collect the specimen in a sterile screw capped container containing 5 ml sterile normal solid semen. Instruct the patient to collect in a sterile wide mouth container. Reagents, consumables: sterile distilled water, conical bottom plastic tubes, sterile pasture pipette, microslides, diamond marker, Ziehl-Neelsen stain, spirit lamp and matchbox, microscope and immersion oil. Equipments: autoclave, biosafety cabinet, bacteriological incubator, centrifuge and microscope. Procedure: sputum pickup, flex of gaseous, necrotic, cheesy, rusty or blood tinged specimen to make uniform thin smear or on a new glass slide. If the specimen is watery, centrifuge at 3500 rpm for 20 minutes and prepare the smear from the sediment on a new glass slide. Bronchospectric specimen: centrifuge the entire specimen at 3500 for 30 minutes and make smear from sediment. Concentration procedure: for urine only, centrifuge the entire urine for half an hour, pull the sediments and proceed with the digestion as for sputum. Fasting gastric aspirate 3 samples. Centrifuge the specimen 3500 rpm for 30 minutes. Prepare a smear from the sediment. First, prepare the smear and stain. CSF: centrifuge the entire specimen at 3500 rpm for 30 minutes and make smear from sediment. Body fluid: centrifuge the entire specimen at 3500 rpm for 30 minutes and make smear from sediment. Tissue: place the tissue in a sterile tissue grinder, then add 1 to 3 ml of sterile saline 1 drop at a time and grind thoroughly. Then transfer the fluid materials to centrifuge tube with the help of pasture pipette and make the smear on new glass lines. Semen: prepare this smear and stain. Mix this sediment well and make the smear from each specimen on individually labeled microsets. Heat fix and dried smear. Stain one smear with Zeton stain. AFB staining procedure: flood the smear with the Zeton cardboard friction reagent and heat at interval until the steam rises for 5 to 7 minutes. This can be accomplished by flaming the slide from below the rack with the spirit lamp. Do not allow the steam to boil or dry out. rinse with the distilled water and tilt the slide or to drain. Decolorize with 20% sulfuric acid with 5 minutes or until the pink color fades. Rinse the slide with the distilled water and tilt to drain. Stain the slide with the methylene blue reagent for 1 to 2 minutes. Rinse the distilled water and allow to drain. Note: for urine, 3% acid alcohol is used as a decolorization. Microscopic examination. Examine the stained slide under oil immersion objectives. At least 100 oil immersion field for presence of AFB. The microbiologist shall observe the slide to verify the results and shall record the result. Interpretation result. The typical acid-fast base layer appears red, slightly curved, short or long rod. They may also appear beaded or banded. Report the results with grading as below. No AFB per 100 oil immersion field, no negative. 1 to 9 AFB per 100 oil immersion field, scanty, record the number. 100 to 10 to 99 AFB per 100 oil immersion field, positive, gram positive. 1 to 10 AFB oil field is positive 2 plus. 100 more than 10 AFB per oil immersion field, positive 3 plus. Quality. Check the quality of the stain with known positive and negative slides.